Now let's talk about some of the candidates for taste receptors. As you can see by this diagram, most of the candidates for taste receptors are G protein coupled receptors. But some of these receptors have a very unusual structure. As you can see, the putative receptors for savory or umami has this very large amino terminus. Likewise for sweet receptors, while the beer receptors look more like a conventional G protein coupled receptor. The best candidate so far for the salty receptors are sodium channels. In this case, sodium channels called ENaC, while for sour, the best candidate is this very unusual type of channel, is a trip type channel. In this case, it's called PKD1 channel. Some of the evidence for those putative taste receptors come from experiments like this, in which there was a gene knockout for several of those different genes, and then the mice were tested for responses to the different flavors of in this case, umami, sweet, bitter, sour, and salt. So what are the conclusions in regard to signal transduction pathway for umami, sweet, bitter, sour, and salty? For the sweet receptors, those are the best candidates for sweet receptors so far. And we think they work as heterodimers, and they are of what we call taste receptor type 1, TRR2, and TRR3. And these two G protein coupled receptor work as a dimer. And what you can see is that the large or long amino terminals form this binding domain that seems to be able to bind a variety of compounds that we associate with sweet. So compounds such as sucrose, fructose, artificial sweeteners, and even some amino acids and even some proteins are able to elicit a very sweet sensation of taste, and all those compounds are able to bind in this domain of this G protein coupled receptors. Mice that lack TR2 or TR3 lose the behavior sensitivity to sugars and artificial sweeteners, again emphasizing that these G protein coupled receptors seem to be involved in the transduction of sweet sensation. The umami receptors, they are very similar to the sweet receptors, and we think that the receptors that transduce the sense of savory are composed of heterodimers of TRR1 and TRR3 to form this what we call the savory receptors. And these receptors bind with high affinity to amino acids that we associate with the savory sensation, and amino acids such as L-glutamate, and also some food enhancers such as IMP that are able to enhance that sensation of savory in the food. And as you can see, some compounds are able to bind to both types of receptors, sweet receptors and umami receptors. While sweet receptors and umami receptors are composed of single heterodimers, the berry receptors are much more diverse. We think there is 20 or more genes that compose those berry receptors, and those berry receptors, as you can see, are G protein coupled receptors, but have a different structure than those G protein coupled receptors involved in sweet and umami transduction. In this case, those berry receptors have a very short amino terminus, and we don't know exactly if they work as monomers or if they work as dimers, heterodimers, but nevertheless, they have this. They don't have that long amino terminal binding domain. Uh, this class of receptors are very diverse because we think it's very important during evolution for the animals to be able to sense a variety of compounds that can be very dissimilar in terms of structure, and they're still associated with of this beer sensation. And we think that these types of receptors can be expressed, more than one type can be expressed in each one of those beer sensation taste cells. The sour receptors is still somewhat unclear what's the molecular identity of the sour receptors. We know that the sour receptors are more sensitive to weak acids such as citric acid or acetic acid, and less sensitive to very strong acids such as HCl. And the reason is that the hydrogen that comes from those acids actually interact with the receptors from the intracellular side. So what the weak acids are able to permeate through the plasma membrane and dissociate and then bind to the to the to these receptors, while the strong acids dissociate from the outside, and therefore they're much less potent. And some of what has been proposed as sour receptors are some epithelial sodium channels that is known to have been modulated by pH. Hyperpolarization activates cyclonucleotide gate channels or HCN channels and acid sense ion channels. But more recently, two members of this trip channel superfamily ion channels have been identified as as taste receptors, and those have this names PKG1L3 and PKG2L1 as perhaps as the major transducers of sour taste. It's not completely clear if they are the only transducers for sour, because knocking out these channels PKG1L3 and PKG2L1 is not enough to remove all the sour sensitivity in behavioral tests for mice. Here is a figure showing that these two types of trip channels are expressed in taste cells and in the mice here, and you can see that these two types of trip channels pretty much co-localize, and you can see here in this fluorescence images here that these two types of channels, the PKG1L3 and PKG2L1, they co-localize, while the TRPM5, which we are going to talk a little bit later, is the major candidate for being the final channel to transduce sweet sensation and beer sensation. They are not co-localized to these trip channels. Some of the physiological properties of the strip channels, the PKG1L3 and PKG2L1, are consistent of this being the SAR receptors, and you can see here some of the electrophysiological property of these channels when expressed in a cell line. And what you can see here, these channels, when 25 millimoles citric acid is applied to these cells, there is this very strong inward current elicited by this acid after a certain delay, as you can see. And here is a control, just the expression vector without the coding regions for these two types of channels. These channels are not blocked by amiloride, and you can see here the dose response in terms of current and pH, that as expected, weak acids are more potent to evoke currents for these channels than strong acids such as HCl. And finally, here is the permeation properties of this channel that reverse about 0 millivolts, and therefore they're able to evoke depolarization into these cells. Salt receptors are also a little bit controversial about which channels or receptors evoke the sensation of salty, and the best candidate, at least in rodents, is the amiloride sensitive sodium channel called ENaC, because amiloride blocks pretty well sodium chloride evoke responses in salt receptors in rodents. Although the controversy here is that in humans, this effect of amiloride doesn't seem to be very strong, so there's still some doubts about the identity of the salt receptors, especially in humans. These different signal transduction pathways and different morphological and physiological types define these two major types of taste receptors, or taste receptor cells, type 2 and type 3. Type 2 that express either bitter or sweet or umami receptors, and those receptors are again activate G proteins that will activate phospholipase C beta 2 that will release a calcium from the calcium store and ultimately lead to activation of tryptophan-5 that will depolarize the cell and lead to release of ATP that will be sensed by the nerve that innervate these cells, and that's the signal that's carried to the brain. In the type 3, on the other hand, which has been so far only associated with the SAR receptors, it does not have a tryptophan-5, and this type of taste receptors seems to be a more conventional type of neurotransmitter release that depends on calcium and release more conventional neurotransmitters, perhaps serotonin or GABA or ATP. Because each type of taste receptor cell express one type of taste receptor, we think that the label line model for the periphery better explain the encoding of taste sensation, at least at the level of the periphery. Finally, I would like to say that adaptation for taste receptor is present, but occurs in a time scale of several seconds. So if you look in a time scale, in this case of 2.5 seconds for the sour taste, you don't see too much of adaptation. So adaptation doesn't seem to be as important for taste receptors as it is for example, for phototransduction or mechanotransduction that we saw in other lectures. So I would like uh, uh, to finish this saying that I would like to know some of the similarities, the difference between the main and accessory olfactory signal transduction pathway, the important role of calcium in olfactory cell adaptation, um, chloride channels as a booster for olfactory receptor responses, the major classes of taste cells and their putative receptors, and the concept of label line in the taste sensory pathway.